Now, genomics has told us quite a bit about the underlying mutational spectrum that gives rise in the landscape, if you will, of tumors. Fundamentally, what are the actual mutations that cause a cell to go from a normal cell to a cancerous cell? The great challenge and the gap, if you will, is that for approximately 60 to 70% of tumors, there is no single driver oncology mutation that's been identified, meaning it's either a mix of multiple mutations or multiple mutations with other changes that actually result in the transformation of the cell. And because of that, it's hard to understand what represents the ideal target for this cancer. When there's a single oncogenic mutation, in the case of KRAS, or in the case of HER2/neu in breast cancer, or EGFR in non-small cell lung cancer, when there's a single mutation, a genetic mutation that gives rise to the tumor, we now have a singular target or a pathway that we can antagonize as part of a therapy. When we look at those tumors, however, for which there is not a clear single driver mutation, and that represents the majority of human cancers today, it's less clear exactly how we begin to approach a therapeutic sort of efficacy in that tumor and how we can precisely target that tumor without actually affecting normal tissues. And this is where the real gap lies today. It's clear that when means are brought to bear, such as immunotherapy, that's not based on a singular genetic mutation, but rather a phenomenon in biology that's present in the tumor, we can have great efficacy in targeting these sort of cancers. But where the gaps lie currently is in understanding for those tumors for which there's not a clear driver mutation. Once again, this represents the majority of tumors. For those tumors, understanding what are the abnormalities that are present, how the tumor has transformed, what are the pathways that are active or not active, and what are the actual targets that are expressed that we may be able to antagonize as part of these precision-based therapeutics, whether it be for ADCs, T-cell engagers, radioligand pharmaceuticals, CAR-T therapies, cancer vaccines, or even standard kinase inhibitors and other more classic chemotherapeutic agents.